Hello, this is Professor Miller. I'm here running a review session for Biology 1406, the Amylase Pilot Lab Units 8 through 12. This corresponds to fall 2019. As we get started, I just want to remind you if you have any questions about things that we're covering or things that we're doing, please go to your lab instructor. They are the best source of information for anything that we've covered here in the lab. As we begin, I want to start off by going back a little bit and thinking about what we did before Unit 8. So thinking about the previous units, the previous practical. Previously, we talked about this following reaction where we had starch, which was a carbohydrate or a polysaccharide made up of hundreds of alpha glucose monomers linked together. We could take that substrate, starch, and expose it to the enzyme amylase. Amylase, remember, is a protein enzyme that speeds up reactions. And what amylase could do is it could break down the alpha glucose linkages in starch into our products of maltose. This was the reaction that we looked at quite a lot previously to Unit 8. What we did when we came to Unit 8 is we began to switch gears and we started talking about a new reaction. It's this new reaction that I want to talk about right now that you should understand really well. So the name of the substrate that we began using in Unit 8 was a substrate called cellulose. Cellulose is a carbohydrate. It's a polysaccharide. It's made up of hundreds and hundreds of beta glucose monomers. This is our substrate. Cellulose, when it's exposed to the enzyme cellulase, remember cellulase is a protein, it's an enzyme, cellulase can break down the cellulase into our product, which is glucose. Glucose are monosaccharides, those are our products. An important thing to remember here about cellulose that is different is that cellulose is made up of beta glucose monomers. This was different than what we were dealing with starch previously. Because cellulose is made up of beta glucose monomers, not all organisms contain the enzyme cellulase that can break apart those beta glucose monomers. We actually find cellulase in some bacteria. We also find cellulase in some fungi. To talk about an organism that is able to produce the enzyme cellulase, we sometimes call these organisms cellulitic or cellulolytic. These are the same term, they mean the same thing. They're describing organisms such as bacteria and fungi that are able to produce the enzyme cellulase. In Unit 8, what we began looking at was dirt. We wanted to see if we could find any bacteria living in our various dirt samples that had cellulolytic activity. In order to do this, we had to run some different dilutions of our dirt. There are two different terms that we talked about that day as we created our serial dilution. We talked about dilution versus dilution factors. Dilution is a value that is going to be less than 1. It's expressed as a fraction or as a percentage. In this case, we were diluting by a factor of 10. And so that meant our dilution was 1/10 or 0.1 or 10 to the negative 1. All of these three terms over here where we're talking about dilution are the same ways of talking about the same dilution. Although we diluted 1/10, you just heard me trying to say it, we had a dilution factor that was greater than 1. So we had a dilution factor of 10 times. What this meant in actual practicality is that we filled up different tubes of 9 milliliters of water, and then we added 1 gram, which is about 1 milliliter of dirt to that sample. This was sort of our initial solution, undiluted. We then took 1 milliliter out of this first tube and moved it into the second tube. So we had 1 milliliter of that dirt water solution diluted in 9 milliliters of water. This was our 10 to the negative 1 or our 1/10 dilution. You did this 3 times so that you got a 10 to the negative 1 dilution, a 10 to the negative 2 dilution, and a 10 to the negative 3 dilution. The reason for diluting this is that there are so many microorganisms that live in the dirt. We were trying to dilute out those bacteria so that we could get a plate where there would be enough colonies that we could then view and analyze. So the purpose for the dilutions was simply that, to dilute the dirt so that we could see a reasonable number of organisms growing on the plate. After you did your cereal dilutions, you then plated these dilutions onto CMC Congo Red growth medium plates. CMC Congo Red plates are a different type of plate that we haven't used before. The CMC stands for carboxymethyl cellulose. The important part to focus in on here is that cellulose. CMC is a substrate. It's a starting material for our enzyme cellulase. The CMC Congo Red really doesn't contain very many nutrients in it. Most bacteria are not going to be able to grow very well on the CMC Congo Red plate because there's just nothing to eat. However, organisms that are able to use cellulose as a substrate will be able to digest that cellulose. In order to see that cellulose being digested, we added Congo Red to the CMC. The Congo Red binds with the cellulose, so if bacteria are breaking apart the cellulose, then we'll see a reduction in the the red color. We talked about aseptic technique that day. That's where we try to minimize the exposure of any of our work surfaces, or especially our bacterial samples, to outside influences. You should know what that word means. In addition, we use the spreader to put the bacterial dilutions on the plate. You should know what the spreader is. We also used parafilm to wrap the plates. After putting your soil samples in the incubator, we had to wait until the next week to look at any results. From there, we then started looking at the unknown bacteria. Each of you were given an unknown bacteria on your bench to work with. With that unknown bacteria, you took that unknown bacteria and 2 other bacteria, and you planted all 3 of them onto a CMC Congo Red plate. Again, remember that CMC plate is important because that is our substrate source for these organisms that may have the ability to produce cellulase. What you did was you streaked 3 different organisms. E. coli was your negative control. E. coli does not have cellulitic activity. It is not going to grow well on the CMC Congo Red plate, and so it was there to show you what negative would look like. B. coagulans was a positive control. We know that B. coagulans does produce cellulase, and so as you streaked it on, you knew that this third of your plate would be able to show cellulitic activity. Your unknown was the exciting one. We wanted to see if your unknown would be able to produce cellulase. Again, we didn't get to see any results in Unit 8. We had to come back the next week to see what would happen. The last thing that we did in Unit 8 was create an overnight culture. In our overnight culture, what you did was you took the cellulase general media, this is just general nutrient growth media for bacteria, and you put in a colony of your unknown bacteria. This allowed that unknown bacteria to grow and multiply overnight, so that bacteria was growing and growing and dividing and dividing and dividing over time. Again, you didn't get to see any results from this. This was something that we then used in the next unit. And that brings us to the end of Unit 8. In Unit 9, we looked at many of the things that we had set up in Unit 8. Just as a reminder, cellulose was our substrate. It is a carbohydrate made of beta-glucose linkages. Often cellulose was provided in the form of CMC. Cellulose, in the presence of the enzyme cellulase, can be broken down into the product of glucose. In Unit 9, the way that we are going to look for glucose production is by doing a DNase reaction. We will also look at enzyme activity by looking at the decrease that exists in substrate. So sort of 2 ways of looking at if our enzyme is working. First, we can look to see if glucose is being produced. That would mean our enzyme is functional. Second, we were also going to look to see if cellulose was being decreased. That would also mean the enzyme was active. So in this experiment, the first thing that we did was we looked at our bacterial dirt plates that we had made the previous week. So as we looked at these, as you looked at the plate, you had colonies throughout the plate, and the plate was mostly red, but around some of the colonies, there would be a clearing where it would be white, and then it would be red everywhere out. This was not as beautiful as this. We'll look at another picture of this at the very end where you can see real examples. But this is generally what you saw. Colonies growing, some of the colonies growing with red all around them, some colonies growing with what we would call a halo. After looking at these plates, what we wanted to do was then calculate two different values. You should be familiar with both of these equations and able to use them. You need to be able to calculate the percent of cellulitic bacteria on a given plate. You should also be able to calculate the CFUs per ml. This stands for culturable bacteria per milliliter in the soil sample. These are two ways to talk about the bacteria. So let's go back to our sort of ugly drawing, and let's imagine we've got 10 different colonies here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, we've got this. So if I'm looking at this particular plate, the way that I drew it is that there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 total colonies. And then we would count up the number that have halos. In this case, it's 1, 2, 3. So to figure out the percent of cellulitic activity, we would say that there are 3 halos at a total number of 10 times 100. This would mean that we have 3% cellulitic bacteria on this particular plate. Note that this example is making a very big mistake. In general, when you look at your plates, you can only calculate percentage cellulitic and CFU per mil when you have a plate that has between 30 to 300 colonies on the plate. I only drew 10 for simplicity's sake, but in real life, we would only want to calculate percent cellulitic or CFUs per mil on a plate that had 30 to 300 colonies. Looking at this plate, we can say that this is our particular plate with a 10 to the negative 3 dilution on it. Looking at that, we'll be able to figure out the CFUs per mil by using this second equation. We calculate up the total number of colonies on this plate. In this case, that was 10, times our dilution factor. Notice it's the factor, so that's the number that's bigger than 1, so 10 to the third. And then we would divide by 0.1, and this would give us our overall CFU per ml, or again, how many cultural bacteria are on that plate. The reason that we divide by 0.1 ml is because this is the amount of media or dirt water that we actually plated on the plate the week previously. After looking at this plate, you should be able to talk about whether or not there were any cellulitic bacteria present in your bacteria sample. The next experiment that we looked at was analyzing your 3-street plates. So in the week previously, you had used a CMC Congo red plate to streak out 3 things. You had your E. coli, you had your unknown, and you had B. coagulans. E. coli most likely did not grow very well for you, if at all. Again, it does not have cellulitic activity. It does not like to grow well on the CMC substrate. For the unknown and the B. coagulans, especially the B. coagulans, what you should have seen is that B. coagulans grew quite well. And what it did was it used up a lot of that cellulose, that CMC. And so once again, you got sort of a halo effect around your B. coagulans. Your unknown most likely looked similar to beta coagulans. Again, it was mostly red around it, but then you had some sort of halo effect. That halo effect allowed you to recognize that your unknown was showing some cellulolytic activity. It was able to produce the enzyme cellulase that was then digesting the substrate CMC. From here, you then went to your overnight culture that you had made of the unknown. In Unit 8, you had taken some cellulase general growth media and you had put in one bacterial colony. You had allowed this to grow overnight, so that bacterial colony had reproduced all night long. And as that bacterial colony was sitting in the cellulase general growth media, that bacteria was producing the enzyme cellulase. So cellulase was being secreted by those bacteria in that media. We know that your unknown was creating cellulase because, again, going back, we had seen on the tri-seq plate that your unknown did have that cellulolytic activity. What we did for you between Unit 8 and Unit 9 was we took your overnight culture, this thing that had bacteria that had been producing that enzyme cellulase in it, and we took this and we spun it down for you. What that means is that all of the bacteria pelleted to the bottom of a tube, and then at the top of the tube, we were left with just the media that contained the enzyme. We then took off just the media that contained the cellulase, and we provided that for you to use. When we did this, just to remind ourselves of some terminology, the liquid that we took off, this is called the supernatant. The supernatant is the liquid that the bacteria was growing in. However, it is not the actual bacteria themselves. What we wanted to do in the third part of the experiment in Unit 9 was to look at this supernatant, this growth media that you had been growing up, to see how active that cellulase enzyme was that had been produced by your unknown bacteria. In order to do this, we set up the following two conditions. We took 1 milliliter of your unknown bacterial supernatant. Again, this is the stuff that we think is making cellulase. Just as a reminder, 1 mil is 1,000 microliters. And you added this unknown bacterial supernatant to a tube that contains CMC substrate. Again, this is our substrate. This is our source for the enzyme to work. You allow the CMC substrate and the unknown bacterial to mix because this would give a chance for the enzyme that was in our bacterial supernatant to then start digesting or breaking apart the cellulose that was in the substrate. In addition to this tube, you also had to create a blank condition or a negative control. In this instance, we added CMC substrate, just like we did in the first case, and then we took just general cellulase growth medium that had no bacteria in it, and so there shouldn't have been any enzyme activity. We took these two tubes and then we incubated them both at 50 degrees Celsius for 30 minutes. The reason for this incubation was to allow the enzyme up here that was in our unknown bacterial supernatant time to digest the CMC substrate. While this was incubating, you then set up a serial dilution of a standard curve, similar to what we had done in Practical 1. We started off with a solution of 15 millimolar glucose. This was the standard that we gave you. You then created a serial dilution, diluting in halves, where you diluted 15 millimolar into 7.5, into half to 3.75, etc., etc. You took this serial dilution, and as you were diluting, you were diluting in waters. Because these dilutions were created in water, this means that you would also need a blank created in water. So at this point now we have two blanks. We have a water blank that's going to be used with our serial dilution, these glucose standards, because those were made in water. And then we also have a media blank. This media blank will be important because this blank that has the media in it is going to be used as a blank for our unknown supernatant. After all of your incubation for the 50 degrees Celsius, Celsius at 30 minutes completed. You then took all samples and you added DNS reagent. DNS is a reagent that is able to react with reducing sugars, of which glucose is a type, and it's able to change colors. We'll look at those colors at the very end. You took the DNS, you boiled it for 5 minutes. After all of your samples had been boiled for 5 minutes, you then analyzed these samples by reading them in the spectrophotometer. Specifically, you set your spectrometer to read at wavelength 540 nanometers. So you put in each of the tubes that you made, you had your standard that you made, and you read the absorbance of the 15 and the 7.5 and 3.75 millimolars and all of your standards. You then blanked the machine using your media blank, because it had media in it, and you read the absorbance of your unknown, and you got some value. After obtaining all this value, that you then created a standard curve and figured out how to figure out the concentration of your unknown. The independent variable, the thing that you were in charge of, was the concentration of glucose. The dependent variable was the absorbance of the different known samples. So what you did was you took 15 millimolar glucose and its absorbance and you plotted it. Then 7.5 millimolar glucose and plotted it. Then 3.7. And you plotted all of your known samples to create a standard curve. Once you created your standard curve, you should have also gotten an equation of a line that would fit the form y equals mx plus b. These equations of the line end up looking something like y equals 3.4x plus 0.042, something like that. With that equation of the line and the standard curve, you then could calculate the concentration of glucose in the unknown solution. There are two ways to do this. One way to do this is just with the graph method or the box method, and this is an approximation. With the approximation, you would do the following. You would read the absorbance of the unknown on the y-axis. Once you find the absorbance on the y-axis, you then go across till you hit your standard curve line. Then you drop down wherever that yellow line crosses the x-axis. That's your approximate glucose concentration. We wanted to be really precise, however, so we asked you to use your equation of a line to figure out the exact concentration of glucose in your sample. In this case, you plug in your unknown absorbance for the y value. So 0.493 is y. And then you plug in the rest of the equation. Equals 3.4x plus 0.042. You then use this equation and you solve for x. So 0.493 minus 0.042. Do that first, then divide it by 3.4, and that will give you x. This concentration of x is your actual concentration of glucose produced in that particular environment. Please note that this equation changes every time you run a standard curve, every time you do the experiment, as one table does it versus the next. This equation will change. So don't just memorize those values, memorize how to use this equation to figure out the unknown concentration. From here, hopefully you were able to find out that your unknown produced glucose, some produced at higher levels, some produced at lower levels, but now you have another piece of evidence that your unknown bacteria is producing cellulase. Unit 10, we continued our evaluation of this equation that should be looking very familiar to you. What we wanted to do in Unit 10 was we wanted to explore better how we could optimize our enzyme, our cellulase activity. Most of us by this point have found that our enzyme is producing some cellulitic activity. However, what we want to do is maximize that. There's a lot of variability in different bacteria about how much cellulase, how much enzyme they secrete. This might be influenced by temperature, by pH, the addition of certain molecules. And so in this Unit 10, you were trying to figure out how to maximize or how to increase that cellulase activity. This is really important in industry because it turns out that we get lots of cellulose or plant byproduct in a variety of different industrial applications. What companies can do is they can take this cellulose, this waste stuff, think like wood pulp or something like that that's left over, and then if they expose that cellulose to the enzyme cellulase, that enzyme can produce lots of glucose, which is a very important end product. We can use glucose for a variety of other applications. So if I'm trying to make lots of money, what I want to do is I want to try to figure out what conditions are going to give me the best cellulase activity. The first thing that we did was we created a bacterial suspension. To create the bacterial suspension, what we did was we took 1, 2, 3, 4 different bacterial colonies from our plate and we swirled it in 1 milliliters of media. This created a mixture where we had multiple bacterial colonies growing, and this was what we were going to use to pull from to inoculate other colonies. This allows us to know that if we are going into new tubes, that we always get exactly the same amount of bacteria into each of the new tubes. Unfortunately, if we had just picked up single colonies and inoculated new tubes, we may have gotten varying amounts. So this was a way of making sure that when we inoculated new tubes today, we were always getting the same amount of bacteria added to each of those tubes so we could then compare them. Each tube had to choose one of 2 variables to test initially. Your variable choices were to test either pH or temperature. Each of you were given tubes that contained 3 milliliters of growth media. You then chose whether or not you wanted to see if pH would influence your enzyme activity or if temperature would. To each of these tubes, you added 50 microliters of that bacterial suspension that you had created. If you had decided to test the effect of pH on your enzyme production, then what you did was you took the samples at the various pHs that already contain these different pHs, and you grew up these samples at 30 degrees to see what would happen. If you were interested in seeing the effect of temperature, what you did was you took your 3 milliliter culture plus the 50 microliters of bacterial suspension and you placed it into one of 4 different temperatures to see what would happen. After choosing either pH or temperature to see how that might affect the cellulase production in your particular sample, you then chose one of 5 different additives to test. You could have chosen any of these 5 additives. Some of these are common cofactors. Cofactors are non-protein helpers of enzymes that are sometimes required by enzymes to increase their efficiency. You were just hoping that maybe one of these would increase your cellulase production. After choosing your additive, you were then given a couple of different tubes. In those tubes, they contained 2.7 milliliters of media. You were being asked to then add 0.3 milliliters, which would be 10% of the volume, of different variables. You should have created at least a 10% additive tube and a 0% additive tube. To create the 10% additive tube, you would have not added any general media, but you would have added 300 microliters of whichever additive you chose to test. For the 0% tube, you would have added 300 microliters of general growth media, but no additive. You would then use this to create other concentrations. I'm not sure what you might have chosen. If, for instance, you chose 5%, you may have used 500 microliters of general media and then 150 microliters of additive. This was the experiment in Unit 10. Again, we didn't really see any results. It was just choosing which conditions we were going to set up and then getting everything into the incubators. Unless you tested variable temperatures, all of the samples that we incubated that day, all of the pH samples and all of the additive samples should have been stored at the 30-degree incubator. That's generally where most bacteria grow well. Unit 11 began with a description of PCR, which stands for polymerase chain reaction. PCR is a technique that allows us to quickly amplify a particular segment of DNA. We're going to use colony PCR to amplify a particular portion of our unknown bacterial's DNA to figure out the identity of our bacteria. To understand PCR, it's a basic three-step process. In PCR, the three steps that we go through include first, a denaturation step. In this step, what happens is the DNA gets heated and it opens up the DNA into two single strands. There's a great diagram of this on page 95 in your lab manual. In the second step, after opening up the DNA, we can imagine that we're at the first step. So first step, our DNA is all stuck together. When we denature that DNA, what happens is it pops open. Once it's popped open in second step, we allow primers to bind. These primers are specifically designed to match DNA close to the segment where we're looking for. So this is a little piece of DNA that I've created that's going to match on either side. So in that annealing step, we allow those primers to anneal to the template DNA. In the elongation step, we raise the temperature, and this allows the enzyme DNA polymerase to actually do DNA replication. Polymerase, same as you've been talking about in the lecture, is an enzyme that allows our cells to copy DNA. So what happens is there's an enzyme that will come along, and it will bind to that primer, then it will copy the DNA. It will bind to the primer, and it'll copy the DNA. So where I started off with one piece of DNA, by the time I am done with a round of PCR, I now have two copies of that DNA. In order to make PCR work, you need a couple of different things. Number one, you need a machine that's going to be able to heat and cool very specifically this sample. That was the PCR machine that we were using. Number two, you have to have primers. These primers are specific for whatever gene it is that you're looking for. We're not copying the entirety of this organism's DNA. We are just trying to amplify a very particular portion of the gene. So the primers are allowing me to look for that very specific area. You then have to include the enzyme polymerase. Again, DNA polymerase is an enzyme that is able to replicate DNA. We are going to use a specifically heat stable polymerase that will not denature when we begin to heat and cool this sample. You also need to add nucleotides, A's, T's, G's, and C's, so that we can build the new DNA strands, and then different buffers are needed. As you ran your PCR, you were trying to amplify a particular gene called the 16S gene. This is a ribosomal gene. All cells have ribosomes. All ribosomes are very similar. However, they do have slight differences. It's been found that we can use this 16S ribosomal gene to tell one bacteria from another. This is a way to identify which organism you're using. The specific region of the 16S ribosomal RNA gene that we're trying to amplify is approximately 1,500 base pairs of DNA long. So for this sample, what you did first was you began with a Cellulase 5% DNA extraction. So you began by mixing some bacteria and TE in a tube and creating sort of this bacteria mixture. From here, you took the bacteria TA mixture and you added it to a tube containing Cellulase. Cellulase is a way of binding to things other than the DNA. You then heated this, you mixed it, and then you took this Cellulase bacteria TE sample and you spun it quite fast, 13,000 RPM for 90 seconds. When this happened, all the Cellulase would go to the bottom of the tube with all the cell gunk in it, and then up at the top you would have supernatant. In this liquid, we would have now some DNA, some isolated DNA from your unknown bacteria. It was this supernatant that we could then add to our PCR reaction. In the PCR reaction, you had a couple of different conditions. In one condition, you had the following. You put water along with the master mix. The master mix included things such as the primers, the polymerase, the nucleotides, and the buffers. Nothing really should have happened in this tube because it just has water. There's no DNA in it. This is just a negative control. In the remaining tubes, in tubes 2, 3, and 4, you added 5 microliters of your DNA that you had just created from your unknown bacteria. So you took some of this, that DNA supernatant, and then you added this to your master mix. Because this has a DNA template in it, we do expect that these samples will do something when they're in the PCR machine. Hopefully these primers will be able to find the 16S ribosomal gene, and they will be able to amplify that 1500 base pair sequence that we're looking for. These samples ran in their PCR machine to amplify that DNA. They were then stored at the freezer until you came back to use in Unit 12. The next thing that you did in Unit 11 was you completed Unit 10. So you took all of your tubes from the previous week. Maybe these were your pH tubes or your temperature tubes, and these were also your additive tubes. And you wanted to figure out how much glucose was produced in each of those conditions, and you were trying to quantify this in millimolar. To do this, you began by taking each of your experimental conditions and putting 1.5 milliliters of that bacteria and maybe pH additive, or maybe it was calcium chloride, and you took 1.5 milliliters of that growth and you spun it down in a microcentrifuge tube. This caused all the bacteria to go to the bottom of the tube, whereas all of the growth medium was there at the top in that supernatant. Again, what we hope had happened is that while the bacteria had been growing, they had been making the enzyme cellulase, and so in that supernatant, you had a whole bunch of cellulase activity. After collecting the supernatants, you then did very similar to what you did in Unit 9. You took a tube, you added 0.5 milliliters of the CMC substrate, right? And this again is our source, that's our cellulose. And then you added, let me make sure that I say the right amounts, 1 milliliters of your supernatant. There it is, spelled right, unlike up here. Ugh. All right. And you allowed this to incubate for 30 minutes at 50 degrees Celsius. You did this for each experimental tube you had. So maybe this was pH 5 tube, a pH 6 tube, a 7 tube, an 8 tube, a 9 tube, a 0% magnesium chloride, a 10% magnesium chloride. And what you were allowing to happen to all of those experimental tubes during this time was you were allowing all of that enzyme, all of that cellulase, to have a time to digest the CMC substrate and produce glucose. We provided for you that day a standard curve of known glucose standards. So while this was incubating, many of you went ahead and ran your standard curve. On your standard curve, you knew the concentration of glucose in the provided standards. You then measured their absorbance at 540 nanometers. And what you did was you created your beautiful standard curve and you got your equation of a line. After you had your standard curve, you then went back to all of your experimental samples that had been incubating for 30 minutes at 50 degrees Celsius. You added the DNS reagent. Remember, the DNS reagent is going to react reducing sugars. It's going to show us how much glucose is in the samples. After you added the DNS, you allowed these to boil for 5 minutes. After their color changed, you allowed them to cool off. You then added water, and you took all of your experimental tubes, and then you ran their absorbances. So you ended up getting data that looked something like this: pH 5, pH 6, pH 7, pH 9, 8, pH 9, and then you got their absorbances: 0.042, 0.156, 0.039, and you collected the absorbances for the different experimental conditions. In order to read the absorbances of these experimental conditions, it was really important that you used a blank. The blank tube that we created for all of our experimental conditions was a tube that just contained cellulose general growth media. The reason that the blank had to have media in it was because all of our experimental conditions also contained media. You used a different blank for your standard curve because your standard curve was made using water. So you had a blank made out of water that you used with your standard curve, and then a second blank that you created yourself made out of media to use with all of your samples that contain media. At this point, you were then asked to analyze your results of these different pHs, these different temperatures, or these different additives that you had tested. It's really important to recognize that absorbance is not the important end result. Using the equation of the line that you created with your standard curve, you should take all of your absorbance values and change those into glucose concentrations that were created. So you can compare pH 5, maybe created 5 millimolar glucose, whereas pH 6 made 32 millimolar glucose. Given a results from an experiment like this, you should be able to describe which pH is the most optimal, which temperature is optimal, and if an additive is optimal. Again, the way that we're going to know that is by looking at glucose production. If something produces lots of glucose, that would be our optimal condition. So in this case, looking at 5, 6, and 7, these numbers that I totally made up, in this case, it looks like pH 6 would be our optimal condition because it had the highest absorbance and that enzyme was creating the most glucose in that particular condition. In Unit 12, we were going to use a technique called gel electrophoresis to analyze the results of our PCR product from Unit 11. We are also going to identify the bacteria by performing a blast analysis. So in the previous week, you used PCR to try to amplify a 1500 base pair segment from the 16S ribosomal unit. What you wanted to do in Unit 12 is you wanted to see if you had actually created this piece of DNA. In order to do this, we used a technique called gel electrophoresis. In gel electrophoresis, we are separating molecules based on size, based on their movement through a gel as we electrocute it. In gel electrophoresis, we create a gel that has holes or wells in it where we can place DNA. What we did was we took the DNA that you had amplified from the PCR and we loaded it into the gels. We also took some DNA that we knew the sizes of. It was called a DNA ladder. DNA ladders are produced by companies so that it will have DNA of very particular sizes. For instance, it might have a 2,000 base pair piece of DNA in there, as well as a 10,000 base pair piece of DNA in there, as along with a 500 base pair piece of DNA. And you'll know the sizes that are in your ladder. We also loaded that to the gel. DNA as a molecule has a negative charge because of the phosphate on its sugar phosphate background. So what we do in gel electrophoresis is after loading the DNA samples to the gel, we then apply a current to the gel. The negative charge goes at the top, the positive charge goes at the bottom, and while this is sitting, the pieces of DNA that are in the wells will begin to move through the gel. Large pieces of DNA have a hard time moving through the gel and they will move very slowly. Shorter pieces are easier to move through the gel will move very quickly and they'll move quickly to the opposite end. And those bands that are in the middle are of medium size. Each of those lines that I just drew represents a DNA fragment. Again, larger fragments will stay near the top of the gel. Shorter fragments will move farther down the gel. In this case, if this was our ladder and we look at the sizes of our ladder, we would know based on this, oh, that first band shows where we were at when we were at 10,000 base pairs. That second band, oh, that's 2,000 base pairs. And then that bottom band, that would be the size of 500 base pairs. You then took your samples at the same time and ran them. In the water sample, again, since we didn't have any DNA to amplify, you probably didn't see much of anything. You may have seen some faint banding near the bottom. These might have been leftover primers. But again, hopefully nothing happened in your water PCR sample. You then looked at samples 2 through 4, where we had added DNA. This is where we were trying to amplify that 16S ribosomal subunit. And in this case, hopefully you saw a nice large band, maybe some leftover products below, but this large band that should have been about 1,500 base pairs big. This was the exciting piece. This showed that yes, you did analyze, you did amplify the appropriate gene as you ran this. In order to see these bands, normally DNA is invisible. So unlike my drawing here where they appear in black, they're not going to automatically do this. So what we do when we pour our gel is we add something called Cybersafe. There's a variety of different ones. This is the one that we happen to use in this lab. Cybersafe is added to the gel so that when we take the gel and we put it on UV light, all of these bands become visible. So when you were first running the gel, most likely it mostly looked invisible. That's because we couldn't see the DNA because the Cybersafe had not yet been exposed to UV light. Because everything's invisible, though, that's potentially problematic. So what gets added to our DNA samples when we load them is a dye. This dye will be visible to the naked eye, and this dye will run near the bottom of the gel. This will allow me to visually look at the gel while it's running to know that my samples are running, even though I might not be able to see the DNA bands until I'm all the way completed with the gel. At this point, if we had been in a research lab, what we would have done is we would have taken out this red square, this 1,500 base pair band. We would have isolated that DNA, cleaned it up, and then we would have sent it for sequencing. You guys are at the end of the semester. You obviously didn't have time to do this, so we did this for you. We took this 1,500 base pair band, we sent it out for sequencing, and then we came back with a DNA sequence of your unknown. So you had your unknown DNA sequence. This was the Word file that included that large number of ACTTGATTCA, or whatever it was. Again, that was the sequence of that DNA band of that 16S ribosomal subunit. Luckily for us, there is a site known as the BLAST site. This is maintained by NIH, National Institutes of Health. And what this allows us to do is it acts like a Google for DNA. It allows us to search a database of known DNA sequences in bacteria to see if we could find our unknown DNA sequence and match it to anything within the database. It was a really pretty straightforward process. You just copied your sequence, you inserted it into the BLAST query site, and then you click search. After you click search, you then got a list of top results. Each of your results had next to it what is called an e-value. You should know how to analyze the e-values. A low e-value is good. So an e-value of something like 0.14 means that it's a really close match. An e-value of something like 1.745, that's a worse match. So the lower the e-value, the better. And what you could see on the organism on that list was the probable identity of your unknown. You should be able to read BLAST search results. Again, you're just looking for the lowest e-value to identify your unknown. Hi, let's go ahead and look at some images that might review some of these concepts that we've seen. Again, this is for the Cellulase Fall 2019 practical review. First thing that I can mention to you, please sleep well before you take your practical. That will help more than staying up late. Also, as you study, please study in small chunks. Don't sit down to study units 8 through 12. Give yourself little amounts. And then third, please practice recalling. After you've studied something, close your notes and practice writing it down, remembering things from scratch, drawing it out on a board. That will really help you when it comes to the practical. So conversions within the metric system, please make sure that you remember this chart. We've still been using it. We've done a lot of conversion, especially between milliliters and microliters. For instance, if you have 1 milliliter, that's the same as 1,000 microliters. Make sure, as always, that you're able to identify all lab equipment that we've used throughout the lab. We've used the spreader, we've had the vortexer, a bunch of different equipment that you should just be familiar with. Know how to read the serological pipettes, whether it's a 1 milliliter pipette or a 5 milliliter pipette. You should be able to read them and understand the volumes associated with the pipettes. Micropipettes, we continue to use them. You got to know how to use them. So for the P1000, it's got the red number up at the top. This is the thousands, hundreds, tens column. So this would be 210 microliters. For your P200, they're all black. You've got your hundreds, your tens, and your ones column. So this would be 21 microliters. And then your P20, it has the red number on the bottom. This is your tens column, your ones column, and your tenths column. This would be 2.1 microliters. You should be really comfortable with this point in the lab at using the micropipetters and understanding their volumes. With enzymes, please make sure that you understand the difference between starch and cellulose. Again, cellulose, I would be able to tell this is cellulose even if it didn't have the label on here because I see these beta glucose linkages. Each of these monomers are glucose. The entire overall molecule is cellulose, which is a carbohydrate or a polysaccharide. With cellulose, the enzyme that digests cellulose is cellulase. With starch, the enzyme that digests starch is amylase. Here's an example of one of those CMC Congo red plates, and you can see some of that bacterial growth. As a reminder, you should have all of your plates wrapped with parafilm. You can see it on the edge of this. You are looking for any haloing. It's sort of hard to see on this particular plate, but maybe there's some light haloing around this one, maybe some light haloing around this one. To analyze plates, you always analyze plates that are between 30 and 300 colonies. Make sure that you can calculate percent cellulitic as well as CFU per mil. As always, make sure that you can tell the difference between dilutions and dilution factors. This would be the dilution, 10 to the negative 3. The dilution factor would be 1,000. Here's one of those tri-streak plates. Make sure that you know which of these is the negative control, which one is the positive control. Make sure that you know how to read them, understand the importance of the CMC Congo red growth medium. Here are some overnight cultures. These are ones that have all completed growing, and in these overnight growth cultures, you can see that they're cloudy. Again, cloudy means that bacterial growth has happened. Here's our good DNS standard. Here's the blank tube, and then we have our DNS of varying concentrations. Note that a darker, oranger, redder color means lots of glucose, and a lighter color means less glucose. This is a serial dilution where we're diluting our known glucose standard in half each time. So we started with 15 millimolar, then we went to 7.5 millimolar. We had to create a blank that was made with water because all of these standards were also made of water. Using these standards, we can then create these standard curves. In our case, it will actually be glucose concentration versus absorbance, but you get the same thing, concentration versus absorbance. And from this, you can then get an equation of a line, and you can use that equation to figure out an unknown's exact concentration of glucose. Make sure that you know how to calculate the concentration of an unknown solution using both the approximation method and the equation of a line. Here we have some sample data here. We could give you something like this and say, what is the optimal temperature for this particular enzyme? And we could see, oh, at 50 degrees, it seems to be working the best. That's where this enzyme has an optimal temperature. Or here's some pH data. Oh, this thing, it works best at pH 9 because that's where we have the highest enzyme activity. The way you were analyzing enzyme activity was, again, looking at glucose production. Here's an example of what a gel electrophoresis PCR result might look like. Over in here in the first column, we have that DNA ladder. Those are the DNA fragments of known sizes. The second lane is left empty. Oh, sorry, is water. Sorry, there is not empty. That's our water. And then we have 1, 2, 3, 4 samples, all containing the same PCR product. So this band that we see that's nice and dark right here, that's that 1500 base pair, 16S ribosomal subunit. These other bands that you see down here, these may be leftover nucleotides, leftover primers, but they're not the exciting thing. With gel electrophoresis, you should know how the charging works. At the top, we are going to always have the negative charge. At the bottom, we'll always have the positive charge. That's because DNA will move towards the positive end. DNA molecules that are larger will stay up at the top. DNA molecules that are smaller will move towards the bottom. Again, we add a dye. You can see some of that dye right here. Also, this sort of blue line right here is some of the dye. Those are visible always when we look at the gels. However, getting this glowing band pattern is only available when we add the SRB safe, the cyber safe. And that's the end of what we have. Good luck studying for the practical. And